So I'd like to introduce our next speaker, who is Bryan. So Mark originally qualified from Glasgow University, and since he's ended up in New Zealand and is currently working in a veterinary practice, Vet South Limited, and where he's head of research and development.
Thank you, and thanks for inviting me here, and thanks for coming to listen. So I'll change tack a wee bit. I'm going to talk about a novel Swede-associated disease that we uncovered 2 years ago now in New Zealand. And just like to thank Shannon and Debbie, who were involved in this process. Got a bit to gallop through, so apologies. I'm going to gallop through it reasonably fast and happy to take questions at the end. So I guess a bit of background in southern New Zealand, our dairy cows go away for grazing. So I'll give you a wee bit of the background of what happens there and why that happens. Talk a little bit about the timelines, and then really what I'm presenting here from an epidemiological perspective is a whole bunch of case histories and some data analysis and some mortality data, and put it all together for a sort of an emerging disease process. So the very bottom, New Zealand's far enough away as it is, but we live at the bottom of a far enough away country, and that picture there is Southland. If you can imagine, the green bits are where we farm, and the rest of it's pretty rough and not really farmable. The sort of kind of not quite so green bits tend to be where cows go for winter grazing, so they move off the pastoral base and they move into sort of higher country or just a drier country. And typically they graze brassicas, so they would graze swedes and turnips and kale. More recently they're into fodder beet. So 2 years ago, so almost exactly 2 years ago, we were getting a few anecdotal reports from farmers saying that they were either disappointed with the condition of stock on a new Swede variety, or that they were losing stock, or that the stock weren't eating them. This new Swede variety was called a herbicide-tolerant Swede, so it was HT Swede for short, so it was developed to withstand herbicide application. On particular, 14th, 15th of July, we had a high number of deaths in cows, and I'm going to talk a wee bit about that later on. And that was the initial, I guess for us, the initial trigger that made us sort of think there's something going on here. We assumed that these cows, they'd been moved from one Swede paddock to another Swede paddock. We assumed that it was acidosis. I mean, most of our cows over winter that die, or we assume acidosis is the cause of death. However, when we post-mortem these cows, and bear in mind there was about 6 or 8 of them, we found a liver that looked like this. And that was a bit of a surprise to us. We thought that's not an acidotic liver. There's something going on here. There's something toxic going on. And that was kind of our first sort of heads up. So just to give you a wee bit of background, Swedes, brassica species, highly digestible carbohydrate. We know that they produce glucosinolates or GSLs, and that's kind of associated with a bitter taste, and there's a whole bunch of animal health issues with high levels of GSLs. The clean crop system that the company had introduced, the HT Swedes, was designed that you could plant these high herbicide-tolerant Swedes, and then you could spray them with a variety of herbicides, and of course you didn't kill the Swedes. So it was quite an attractive proposition for farmers. It was kind of easy maintenance, quite expensive. So these Swedes are 4 times the price of ordinary Swedes, and that's one of the herbicides there, Telar, that is used to spray on these Swedes. So what happened exactly? So we were getting these kind of sick cows and deaths, but no more than we would normally get over a winter, they were just sporadic. And any of you that are in practice, you get this kind of sporadic stuff happening, and sometimes it's hard to piece together. But occasionally we did get the odd unusual pathology or strange biochemistry or whatever. But it became quite apparent after this initial autopsy of those 6 cows that when we went through the data and we emailed out to the rest of our vets, it became quite apparent that there was one common factor, and I think of 40 farms, 39 of them were associated with this HT Swede. So initially, our first approach was to think, well, we've found the problem here. We'll meet with the Swede producers and discuss it with them. And as you can imagine, that didn't go down very well. And I sort of put up there, we had a divergence of view. It would be akin to sort of, trying to, the best analogy was when you're talking to tobacco companies about lung cancer. It's everything else but the tobacco. So, well, that's all right. We thought, well, we need to study this. We're still in the middle of winter. We need to study this, and we proposed a longitudinal study. Suffice to say that nobody accepted that was a good idea. So then we sort of ramped it up a bit, and we advised Fonterra and MPI, which is our DEFRA, and we started to do a bit more agitation. And what we did was we got all the vets together in Southland and started to compare notes. And the ultimate outcome of that was we formed a sort of a working group, an advisory group, and we got the Swede manufacturers and Dairy NZ and everybody in the same room together, and we sort of moved forward from there. So I think that was quite a significant step. And out of that, the Swede manufacturer did comment that HT Swedes are majorly affected, and that was the first acknowledgement, but this is kind of in mid-spring. That was the first acknowledgement that there was actually some issue there. This is what we proposed when we sat down with these guys from an epidemiological perspective. There's a whole raft of issues here. There's a timeline going on here. We're somewhere in the middle of winter. We're seeing issues around transition onto Swedes. We're seeing photosensitivity. There's obviously a bunch of factors here: Swede variety, the management of these Swedes, the climate. The weather was quite warm 2 years ago, and then there's a whole bunch of issues. And we said, well, we need to really investigate all of this to really understand what on earth's going on. It may well not be the Swedes. That was rejected, and instead, what was happening at an industry level was Dairy NZ picked up a piece to do a retrospective survey out here. So it was kind of, that was the industry response, and we felt it was behoving on us to do a little bit more than that because, as you can see, there was a lot of stuff that was kind of missing. So what I'm going to walk you through now is just some of the stuff that we pieced together. So, and these are just examples because we had a lot of stuff going on, as you can imagine. I think by the end of it, we had about 120 farms affected with this. So, for example, acute deaths. These were cows in one group that were on Kale for 4 weeks, so they had been well transitioned onto a brassica crop, and they, without incident, no problems. They moved on to an HT crop, and within 96 hours, 6 were dead. Again, the presumptive diagnosis is acidosis, but by the next day they've got more deaths, and then again we found this hepatotoxicity. And interestingly enough, even after that and reducing the intake of Swedes, we were still losing cows up to a month later. And the other interesting thing about this disease was other mobs on adjacent paddocks were unaffected, so it was fairly complex. And then we had a whole bunch of sick cows. So the sick cows, here's an example here, a group of cows grazing on HT Swedes for 6 weeks. A number of them had been sick, and quite a number of them developed photosensitization. You get the odd cow going down and a bit of diarrhea, and the farmer would report that they just all look not very good. When you run some bloods on these cows, you can see that the biochemistry is kind of all over the shop, but consistent in that biochemistry is very high GGTs and GLDHs. So this is indicative of a liver issue. There's a bunch of other stuff going on here. When we reported this to the Swede company, their comment was that GGTs of 3,700 were not particularly high. The definition of particularly high is obviously different to mine. The photosensitivity issue seemed to be particularly bad with heifers. So we were seeing maiden heifers on Swedes, on HT Swedes. A number of them were picking up photosensitivity. And interestingly enough, some of these would die, but a lot of them would be okay, and then they would sort of recover, but then after calving, they'd get photosensitivity again. When you ran bloods on these, here's a group of these, again, you're getting a similar sort of picture with quite elevated GDTs, a bunch of other stuff going on. These are fairly complicated blood pictures going on there. And what I want to think about is now transition cows. You've got to bear in mind we're moving through the winter, piecing this together, thinking that there's a common thread here. And our biggest challenge was around the transition cows, and we know that transition cows are obviously the ones most affected. And there's kind of, what I would say, there's kind of two groups here. So there's a group of cows that at winter, as dry cows, because we're seasonal calving, were fed Swedes. And at the transition period, they were taken off Swedes and fed on grass, and then transitioned onto the normal pasture. And a number of those cows went down with milk fever and metabolic disease. and we had a much higher mortality rate of those cows, even though they had been removed off the HT Swedes. There was another group of cows, or subset of cows, for example, that never received HT Swedes over winter, but when they were transitioned back onto the dairy platform, might have transitioned back onto HT Swedes. And that was quite spectacular. We had lots of cows going down with metabolic disease. We had a much higher mortality rate, photosensitization, and a whole bunch of disease. But as you know, when you're working through these sort of processes, it's quite hard to pinpoint exactly what's going on. You're getting reports from many different farmers and many different situations. We did try and run some bloods on cows, and I'm not going to spend too much time on this, but looking at, for example, the difference between Springer cows, Colostrums, and milkers by their history. So have they been exposed to HT Swedes? Have they not been exposed to HT Swedes? And is there a difference? And I'm not going to spend any time on this. I'm happy to go over it with somebody else later on. But as we work through whether there be Springers or colostrum cows or milkers, the exposure or non-exposure to HT had often highly significant biochemical effects, even if we didn't see any disease in those cows. And then here was an interesting case history with a group of cows grazing HT Swedes alongside a group grazing Kale. So almost identical. The paddocks were identical in soil type and aspect, and they were getting very similar rations. And when we blood tested these cows, here's the key results here. You can see the difference between the cows on Kale in blue and the cows on HT Swedes in red. And so again, a lot of circumstantial evidence that this type of Swede is doing some damage to cows. Finally, Youngstock. Towards the end of this process, farmers are getting quite nervous about these Swedes, and there was advice coming out that, well, you probably shouldn't feed it to adult cows, you should feed it to Youngstock, because that's all right if you kill them, And so we had a farmer who had an awful lot of these Swedes and decided to put his Youngstock on. We were uncomfortable with that, but we thought, well, we might as well get something out of this. So let's bleed these calves before they go on. Let's bleed them after 5 days, and let's bleed them 10 days later and see what happens. As it happened, after 5 days, he pulled them off because the calves just looked appalling. And here's the blood results pre, during, and post GGT, GLDH, AST, and CPK. So we're getting a lot of liver damage, we're getting a lot of muscle damage. These calves are not doing very well on this novel Swede variety. The final piece of the puzzle for us, and this is probably has been the most effective piece of the puzzle, was whilst Dairy and Zed were doing their retrospective survey, that was kind of around the transition period, we were quite keen to get really good objective data. And so we thought, well, mortality rates, death rates over winter is probably the best data, the best, most reliable data we could get. So we're not asking farmers what happened, how many cows were sick. We just simply wanted to know how many cows did you send away to grazing and how many cows came back. So you send away 120 and you get 118 back. Then we go back and find out when did they die. Let's work out the specific number of cow days on each crop by grazing, and let's work out the mortality rate on each of these various crops. And so we did that. We calculated the mortality rate on the crops. Now, the way we did it schematically is like this. If you can imagine, you've got a herd here, and you divide them up at drying off, and they go to various grazing. Some of them might be grazing for 2 months here, and there might be a smaller mob grazing for 6 weeks here. And it's not as simple as it would appear in your head. And then there's death through here. And then they come out back to the milking platform, and this is when it gets really messy. We didn't want to touch the milking platform because you've got cows coming in on HT and cows not getting HT and springers and whatnot. So we were just really worried about cows on grazing, mortality rate of cows on grazing. And even that simple schematic, in reality, when we went down and mapped this out and talked to farmers, it looked like that. And so, collecting these data are actually quite challenging. The key thing is that what did we get? We had a lot of cow days. So you can see there we've got 463,000 cow days. And here's the mortality rates per 100 cows per 100 days. Unadjusted odds ratio, 6 to 7 times the mortality rate on HT Swede as opposed to anything else. So, we were fairly comfortable that there was something going on here. If you run a bunch of more complicated stats, so obviously these animals are clustered by group, and you run a GEE, you get very similar results. So here's the adjusted mortality rates. And yes, the error bars are quite big, but the p-value is 0.001 or something. It's highly significant. So winter crop had a highly significant effect on mortality rate. So we were fairly comfortable that HT Swedes was causing this higher, mortality. Been through some of that sort of stuff, and evidence of not only higher mortality, but increased disease and photosensitivity and whatnot. Just one final piece, the Dairy NZ study, which was published after some of our data, they confirmed statistically significant association between disease and HT Swedes in late season and early spring. And subsequent to that, the company then produced a product endorsement which says at the bottom there, we shouldn't feed this to pregnant cows. That's now been changed to pregnant or lactating dairy cows, which doesn't leave many cows left, I don't think. I'll probably run out of time, so I'll just quickly flick through that and stop there for any questions.
Very much. Okay, have we any questions for Mark? Must be, yeah, no, Peter. We have at the front here. Coming behind you.
Thanks a lot. Peter Maloney, Ireland. I suppose you weren't able to find out from the manufacturer of the HT Swede where the gene came from that they inserted to give the resistance.
Well, they're not genetically modified. So
how did they become resistant to
Well, we don't, because we're GM free in New Zealand. So, and this is actually an interesting point. There's a strong argument that if you genetically modified them, you would just insert the resistant gene in there, and that might have been the end of it. They didn't do that, of course. They took a whole bunch of different types of Swede varieties and mashed them up in a vat of weed killer and worked out which ones survived. And it just so happened that the ones that survived also killed cows. So it was a very sort of rough and ready way of selecting for Swedes. So in actual fact, ironically, it's a good argument for genetic modification.
Owen Collins, Chagas. I was just wondering, do you have any record of pesticides that might have been used on the Swedes before the cows were put out? You mentioned that the HT resistant.
So they use Telar, which is chlorsulfuron or something, and they used that prior to twice, I think, before the cows had grazed on it. But I didn't show that data, but I think the LD50 for cows is 2.5 kilograms of Telar, and it's just not conceivable that that the weed killer would have been a risk, I don't think.
Okay, thank you.
Okay, we're going to have to move on. Thank you very much, Mark. Very interesting.